This is podcast 34. So, in podcast 32, we talked a lot about what happens during prophase 1 and how you can break prophase 1 into these different subphases: leptotene, zygotene, pacotene, diplotene, and diakinesis. But we didn't talk more about there is a structure and there's a zipper setup sort of thing. I just want you to know that it is known what this structure is made out of. So in your book, it talks about there being a lateral element and a central element. These proteins have been identified, what make up the lateral element and what make up the central element. And they've been cloned and sequenced, and we know what they are. I don't necessarily know that you need to come up with the names of all these sort of things for this class. I just want you to know it is known. Also, it is known when these start to polymerize and how these start to polymerize. So, for example, SYCP1 in yeast is called ZIP1. It forms this sort of zipper structure that make up a large part of this central element, zipping the two chromosomes together during zygotene. Okay, and then allows crossing over to happen during pacotene because they're in such close proximity to each other. And this, if you think about it, it has to be this close proximity to each other, because if the two chromosomes are misaligned, then you can't have crossing over events, or if you did, it would be disastrous because you would lose a lot of DNA if they weren't perfectly aligned up. Okay, H5P question for you. Of course, mistakes happen, and you can have non-disjunction events where the bivalents do not separate in meiosis 1, anaphase 1 of meiosis 1, and instead both go to one side. That can actually happen in meiosis 1 with the bivalents, or in meiosis 2, where the two chromatid chromosome doesn't separate into two separate chromosomes in meiosis 2. But meiosis 1 non-disjunction is more common than meiosis 2 non-disjunction. In the meiosis 1 non-disjunction, where both of the bivalents go to one cell, what happens is you end up with gametes that have more than they should. They're more than they should have in terms of complement of chromosomes. They have the haploid number plus one extra chromosome, and also gametes that have the haploid number and missing one of the chromosomes. This is an abnormal number of chromosomes in the gametes, and this is called aneuploidy in general. Let's see. This is the point of this one is just to show you that non-disjunction can happen also in meiosis 2, but it's more common in meiosis 1. If it happens in meiosis 2, you can also have aneuploidy in the gametes. Probably the most well-recognized example of aneuploidy is trisomy 21, which causes Down syndrome. Down syndrome is not the only type of aneuploidy in humans that is known. Here's an example of some other aneuploidies. So here's Down syndrome, where you have 3 copies of chromosome number 21, or trisomy 21. This happens in 15 of every 10,000 births on average. There's Edwards syndrome, which is trisomy of chromosome 18, 3 in every 10,000 births. Patau syndrome, trisomy of 13, 2 in 10,000 births. Turner syndrome, which is one copy of X and no Y. This is every 2 in 10,000 births. There's Klinefelter syndrome, where you have two copies of X and one Y, or XXY. This is 10 out of every 10,000 male births. There's trisomy of X, trisomy X or triple X, 10 in 10,000 births, and XYY, 10 in 10,000 male births. Of course, you can have other sorts of chromosomal problems where you have an inversion in part of the chromosome, or a bit is missing or added on. This is only talking about intact extra copy of a chromosome, or actually in this case missing one of the chromosomes, missing an X. So here's an H5P question for you. Okay, in human females, meiosis doesn't quite play by the rules that we've talked about in terms of you go through meiosis 1 and then you might go through meiosis 2. So human females will develop inside their mothers and start developing gametes in their mothers at about 11 to 12 weeks of gestation, but then those eggs stop in prophase 1. So they start going through meiosis, stop in prophase 1, stop shortly after birth, and then they're held in prophase 1 until puberty. At puberty, at roughly monthly intervals of ovulation, an egg is released, and it will progress to metaphase 2, but only proceed past metaphase 2 if it's fertilized. And if it's fertilized, it'll go through anaphase 2, and then the fuse with the sperm nucleus, and then complete meiosis after that. So this is a chart from a different site, not your book. So before birth, diploid germ cells proliferate, then differentiate. So you get cells that are going to become eggs. And you get these primary oocytes. And they will arrest in prophase 1 shortly after birth. And then once a female reaches puberty, all the way through menopause, approximately once a month, one of the primary oocytes will resume meiosis from prophase 1, goes through metaphase 1, and continues through metaphase 2, meiosis 2, but not all the way through meiosis 2. It'll arrest in metaphase 2. And it'll rest in metaphase 2 until and unless it's fertilized. Okay? And then I also want to point out here that during meiosis of human female eggs, you have unequal cell division. So it's not that all cells are dividing completely in half, okay, after metaphase, or after meiosis 1. One cell will get most of the cytoplasm, and another cell will get very little bit of the cytoplasm, and that's called a polar body, which will degenerate. So I asked you earlier in a podcast why have unequal cell division. This is one of the reasons why. You want to concentrate all of your resources in one cell and not divide them among 4 cells like in generating sperm. Okay? So the the primary oocyte will resume meiosis when it's ovulated, and then goes through metaphase 1, continues through meiosis 2, but stops in metaphase 2 of meiosis 2. And then here's yet from another book, because your book doesn't handle this very well, I don't think. So picking up here, okay, egg arrested in metaphase of meiosis 2, metaphase 2, okay. When the male sperm penetrates the egg, only the pronucleus goes forward, right? Remember, you don't get any mitochondria from the sperm, so just the pronucleus goes forward. And the egg will move past metaphase 2 when this happens, and during anaphase 2, will move a set of chromosomes to a second polar body, and then it will fuse the male and the female pronucleus together to form the embryo, the zygote, which then starts going through mitotic cell divisions. So pretty complex, but let's go back here. So in meiosis, we'll start in human females while they're still in utero, 11 to 12 weeks of gestation, then stop in prophase 1 shortly after birth and stay there through puberty. At puberty, approximately each month, an egg will be ovulated. And after it's ovulated, it'll proceed from metaphase or meiosis 1 through meiosis 2, but not all the way through meiosis 2. It'll arrest in metaphase 2. And then if it's fertilized, it'll proceed through anaphase 2. Then the sperm nucleus fuses with the egg nucleus, and then it will finish off meiosis. So you generate, females generate germ cells in utero. We'll make a primary oocyte that is arrested in prophase 1 after the female is born. It'll stay in prophase 1 through puberty. Once you hit puberty, upon ovulation, you have unequal cell division, okay, going from meiosis 1 to meiosis 2, and leaving one set of genetic material behind. Remember, you're going from diploid to haploid, going from meiosis 1 to meiosis 2. And that set of genetic material will degenerate in a polar body, a cell that is a cell in name, but not in function, because all the good stuff went to one of the oocytes, okay, secondary oocyte. Now this secondary oocyte is going to proceed to metaphase 2, and then if and only if it gets fertilized will it go past metaphase 2. If it gets fertilized here, okay, then it'll go through anaphase 2, put that set of chromosomes, half of this 2-chromatid chromosome set, and put that into a second polar body. And now, after that happens, now the egg is truly haploid, and that pronucleus can fuse with the male nucleus to make a diploid zygote, which will then go through mitosis to develop the person. Okay, so you can have non-disjunction events that happen during development of these eggs, and they are more common as the age of the mother increases. So you have, for trisomy 21, this one particular non-disjunction event, the chances of that happening at age 30 are on average 1 in 1,000. But by age 50, 1 in 12. So much higher chance of Down syndrome in older mothers. So why is that? This is a review that I found. It's a bit dated, 12 years old, but the best review I could find. And in that review, they said that it's possible that in an older woman, this egg has been arrested in prophase 1 from her birth um, for 50 years. And then it goes past prophase 1 upon ovulation. So in those 50 years being arrested in prophase 1, remember where that's where synapsis happens, some bad things could happen, okay? That could cause non-disjunction. 90% of the time, the non-disjunction is in maternal meiosis problems versus a paternal meiosis problems. This is in the case of trisomy 21. Most of the time, it's going to be meiosis 1 versus meiosis 2, as I showed you before in that diagram. So both of the bivalents go to one side versus the other side, meiosis 1, in anaphase 1 of meiosis 1. And within those cases, most had no crossover events for chromosome 21. So a question might be, well, maybe something happened because it's sitting in prophase 1 for so long. Maybe something has happened that in the crossing over that contributes to this non-disjunction. And the opposite seems to be true in that for most of the cases where there was trisomy in chromosome 21, there hadn't been any crossing over in 21. So that does not seem to be related to why this is happening. Men aren't off the hook either. There is a hypothesis that men who have kids as fertilized kids anyway, as older men, they may have an increased risk of autism in their offspring. There are data to support this. There are data that refute this, and safe to say that we don't know all the causes, just like we don't know all the causes of maternal non-disjunction.